Okay, our last topic for Chapter 6, we're going to talk about a little bit more about growth of bacterial populations and some additional terminology and so forth associated with this. Your textbook provides some more details on this, including some mathematical details that we're not going to really get into, so we're just going to kind of hit some of the highlights here, some of the important info. Okay, bacteria multiply or replicate through a process called binary fission. Binary meaning two, fission to split. So one cell splits into two cell, one cell to two cells. That's binary fission. Now, when you study human cells, you learn about a very complex process called mitosis, by which a eukaryotic cell like ours will divide into two cells. Bacteria don't do mitosis. They go through a process called binary fission, which is not quite as complex. The reason it doesn't have to be quite as complex, or the key reason if you think about it, how many chromosomes did we say most bacteria have? Just one. What about your cells? Your cells have 46. So when one cell divides into two, one human cell divides into two, guess what? Both daughter cells have to have 46 chromosomes. And those chromosomes, you've got two copies of chromosome number 1, two copies of chromosome number 2, two copies of chromosome number 3, all the way up to 22. And then you've got two X chromosomes, or you've got an X and a Y if you're a male. When one of our cells divides into two, you've got to copy all 46 chromosomes, and then you've got to make sure that each of your new cells over here gets two copies of number 1, two copies of number 2, two copies of number 3. You can't just randomly send those 46 chromosomes into each of the two daughter cells. You've got to have just the right ones. The mitosis process is concerned with that, making sure that each daughter cell is going to get two copies of each chromosome. So that's a much more complex process. If you're a bacterial cell and you just have one chromosome to begin with, it's not as complex. You just duplicate it and then make sure each of your daughter cells gets a copy of that one chromosome. So not quite as complex of a process. Okay, so your major steps, as you see, replicate the DNA. DNA that consists of your genetic blueprints. A cell can't operate. A cell's going to die if it does not have its DNA, its genetic blueprints. And that's what you're seeing here on our little chart over here. In purple, that little squiggly thing, that's supposed to be the chromosome of the bacterial cell. It gets copied. Each copy gets pushed to one end of the cell. Then a septum is another term for wall, or a division, begins to develop between those two copies of the DNA. So you've got to create cell membrane and cell wall. Outer membrane, if you're a gram-negative bacterium, you're going to need a new outer membrane as well. So you've got to create a separation between the cells. Once those two cells are completely separated, you now have two cells, the two daughter cells. So one parent cell becomes two daughter cells. All right, and then what they're showing you here is each of those daughter cells here can now undergo cell division. And so 2 becomes 4, and then 4 becomes 8, and then 8 becomes 16. Notice too, like they're showing you here, those 4 cells are sticking together in a chain, and then eventually they're forming a cluster over here. Among bacteria, some bacteria, when one cell divides into 2, the cells immediately split apart. So they don't stay together in a chain, they don't form an actual clump. Other things like your strepto-arranged bacteria, as the cells divide in one plane like this, they stay connected to each other. So they form a chain. Staphylo arrangement, the clusters, like with Staphylococcus, as the cells divide, the new cells are forming in this direction and then also in this direction as well. So they're going to form a 3D clump or cluster of bacterial cells, and that's what we call a staphylo type arrangement. But not all bacteria do that. Some bacteria separate from each other immediately after they divide. Notice the timing over here. 30 minutes, 60 minutes, 90 minutes, 120 minutes. If it takes 30 minutes for one cell to divide into two, that's called the generation time. So after 30 minutes, starting with 1, you're going to have 2. 30 minutes later, you're going to have 4. 30 minutes later, you're going to have 8. 30 minutes later, you're going to have 16. Those times are called generation times. And those times will differ for different types of organisms. Hang on here. Okay, I was having a little bit of a problem here with my iPad that I'm drawing on. Okay. Okay, generation time, as I was just mentioning, that's the amount of time it takes for one cell to divide into two. So for microbes, especially for bacteria that just have one chromosome, that generation time can be pretty fast. So it can be pretty short because they've got one chromosome to divide. The cells are small, much smaller than eukaryotic cells. So it doesn't take quite as much energy and effort for one cell to divide into two as it does for like a human cell, which is much larger, more complex, and has 46 chromosomes that have to be copied. So generation times tend to be short for bacteria. Now there's a lot of diversity out there. There are some bacteria that multiply really, really, really slowly. But in general, the ones that we're interested in studying, the ones that impact the human body, can divide pretty quickly. So generation times are short. If the shorter your generation time, the faster you're growing. All right, now let's think about like if you set up a broth culture of some bacteria in the lab. If you put just one cell into that culture, which would be difficult to do, and that cell, let's say that this bacterial cell is happy, it's got all of the nutrients it needs, it's growing as quickly as it can. Starting with one cell, one cell becomes 2. 20 minutes later, you now have 4. 20 minutes later, 8. 20 minutes later, 16. After just 7 hours starting with one cell, if you kept building those numbers, you would have 2 million cells. And if you could keep providing optimal nutrients, if you could keep taking away the wastes, all cells generate wastes, even bacteria, which would become toxic, you could wind up with 10 to the 21st power cells after 24 hours, starting with just one in your culture. That's a 1 with 21 zeros. If optimal conditions were met. When you have a little tube of broth over here, no, you're not going to be able to meet those optimal conditions in a closed tube of broth from which none of the bacteria can escape, and neither can any of their waste products. Plus, there's a limited amount of nutrients in there, so you're not going to have indefinite ongoing growth in a culture like that. Nevertheless, when I tell you guys in the lab, you don't need to scoop tons of bacteria off of a colony on a plate to set up a new culture. I'll be telling you that shortly. And this is why the bacteria can grow so quickly. You don't need a whole lot of them to start a culture. They're going to very quickly grow into a very large population. When you guys are working in the lab and you see a broth culture like this that looks cloudy, there are at least trillions of bacterial cells within that culture, if not more. Okay, microbial growth in the laboratory when you're growing microbes, especially bacteria here. Bacteria are the main group that we're talking about at this point. If you had a culture of bacteria in a liquid, if you could sample, or if you were, and I won't say could because you could do it if you wanted to, sample some of the bacteria out of that culture and count the number of bacteria that are in there over a period of time, and graph the population. So over here on the y-axis, this is representing our population of bacterial cells. And notice this graph, like where it says 2, that means 10 to the second power. 4 means 10 to the fourth power. 10 up here means 10 to the 10th power. So a 1 with 10 zeros on it. So we're looking at big increases in population here on this graph. All right, on this graph at time 0, here, this is where you first start a culture of bacteria in the laboratory. You started incubating it. Notice that for some period of time, the population wouldn't really change that much. That's called the lag phase of a bacterial culture, where there's not a lot of change in population. And that's because the microbes are probably having to adapt to the new culture medium. The nutrients in there may be different, so they've got to adapt to it. The physical, environmental factors may differ. Also, your textbook discusses this as well, the types of methods that are used to count bacteria. Early on in a culture, they may just not really be detectable by those typically used methods, and that's why you'll see this lag phase. But after a few hours, generally, the microbes will have adapted to the culture, and they're going to start growing explosively. That's called exponential growth or logarithmic growth. Those are mathematical terms that basically refer to a very rapid increase. Now, eventually, you're going to hit a phase where the population doesn't change much. That's called the stationary phase. So for some period of time, it's going to differ between the species. Population is going to kind of hold steady. That's called the stationary phase. Some people call this the plateau phase as well, because it looks like a plateau where it's flat on. You've got like a hill or a mountain and it's flat on top. That's called a plateau. So what's going on there? Well, here in the stationary phase, yes, you do have cells dividing, creating new cells, but lots of cells are starting to die in the culture. Your nutrients are being used up. Wastes are building up in the culture, and the poor little bacteria in the tube can't escape those wastes, so those wastes start becoming toxic. Then if you just kept incubating this culture, eventually you would hit what's called the death phase or the decline phase, where some cells are still alive, are viable, but other cells are dying. A lot more cells are dying than new cells are being created or are remaining viable, so your population starts to go down. So this is typically what you would see, and when we study disease stages later in the semester, you'll see that the populations of microbes, when you have an infection, follow a similar pattern when they're feeding off of the human body as well. So in the laboratory, I'm going to, you know how I've been storing your Petri dishes in the refrigerator, the ones that you guys did from your original swipes? I was sticking them in the fridge. Or when we do the unknown bacterium project, I'm going to have you store those cultures in between class sessions in the refrigerator as well. You don't want to keep putting an older culture back into the incubator, because if you do, if you keep incubating it, early you're going to be in this nice exponential growth phase where the bacteria are nice and happy. But it doesn't take a whole lot of incubation before you wind up over here in your death phase. And so if you just keep incubating, most or all of the microbes in your culture are going to die. So if you put it under refrigeration, so you're below the minimal growth temperature of your organism, your microbes can hopefully stay over here in the stationary phase or maybe early death phase over here, and they don't keep proceeding downward. So you're slowing down the metabolism or stopping their metabolism so that they just kind of hang out in a state of suspended animation, and you can keep your populations in that culture up here for some period of time. All right, so just to review, clinical importance. Why is understanding how bacteria and other types of microbes grow important? When we study disease in more detail later in the semester, as I mentioned, microbes cannot make you sick unless they can grow and reproduce inside the body. So we need to understand the requirements, the environmental factors that are important for their growth. Where in the body can they survive? Where out in the environment can they survive? That's important for controlling microbes as well out in the environment. When are you more likely to shed live microbes and transmit them to others? Where? From what parts of the body are you going to be emitting these microbes? Lab diagnosis, as I've already talked about, if you're going to grow microbes in the laboratory, you have to be able to understand their needs and requirements. so that you can grow them to large numbers and study them. You have to keep in mind that some are fastidious, which means they're picky. So you've got to know, well, what type of culture medium, what do I have to provide in the culture medium? What type do I need to grow this particular organisms? A lot of drugs and antimicrobial control agents, which we'll be talking about in Chapter 9, really work best with actively growing microbes. So that's another important reason why understanding microbial growth is important. This is huge. Again, understanding the parts of the body where microbes live and where they don't typically live. What types of body fluids are likely to contain microbes? That's very important as a healthcare worker when you're collecting samples. And we always emphasize aseptic technique. You cannot, a sample is no good anymore if you've contaminated it with your own skin bacteria, or you're collecting a blood sample and you contaminate the sample with bacteria from the patient's skin. You have to be aware of where microbes grow in the body as well. Very important from a clinical standpoint. All right, so that's actually going to conclude our Chapter 6 information. In Chapter 7, the next video lecture clip I'm going to do, we're going to start talking about Chapter 7, which is going to be about metabolism in a little bit more detail. How do microbes obtain their energy from chemical nutrients and then make use of that energy? So that'll be the subject of Chapter 7.